I think we'll move on to the last talk of the session. Again, there will be time for more questions to Dr. Eberl afterwards in the last 15-minute question and answer session. The last talk of the session is from Professor Germano Iannacchione from Worcester Polytechnic Institute. The title of his talk is Emergence of Order and Disorder Far from Equilibrium, Fundamental Frontier in Thermodynamics and Condensed Matter Physics. Professor Iannacchione.
Thank you all. Can you hear me well? Am I coming through okay?
Yes.
Thank you. Thank you. I'm going to now start my slides. Go now. Here we go. There we go. Well, I'd like to thank the organizers of this wonderful conference. I've enjoyed all the talks immensely, and a special thanks to Ram Padel and Georgie Georgiev, who've been very, very kind in inviting me to give this talk. I'm going to draw upon my experience both in my research, but also as a program director at the National Science Foundation. So my talk is going to be broken up into three parts. One, I'll begin with setting the stage and simply reiterating a lot of the things that have been brought up under many different contexts. I'll then follow that up with a simple example from my research, and then I'll end talking about navigating the research space in general. So as part of my introduction, I'll just simply reiterate some of the main questions that have emerged that we're all struggling with, and what is the foundation of this wonderful conference. Very basic questions, and my perspective here is to look at physical systems because of advances in technology in a new way that may give us some insights, and to lay out that we are already exploring some really important aspects of emergent order from an engineering perspective. So to begin with, I'd like to, again, just reiterate some of the things, just to, as I said, setting the stage about equilibrium thermodynamics. The typical thing I tell my thermo class is consider 1 mole of molecular hydrogen. It's about 2 grams, that's it. But it has 5 times 10 to the 24 numbers that need to be specified in order to uniquely define each and every particle. 3 components of momentum and 3 components of position, plus the volume of the space it occupies. But if you wait long enough and things are steady state and time independent, this distribution becomes homogeneous and can be completely characterized by a much smaller set of numbers. And that's been said in many of the other talks. This opens up what is the real information content of a simple thing like a 2 gram amount of gas. From a thermodynamic point of view, the key features are it's ergodic. If I were to take a series of snapshots and I looked at different regions of space and kept track of the particles, a particular region in space, if I time averaged it, would give me the same average values of energy, average values of momentum, as if I had spatially averaged all of the particles at a single instant in time. So as I said, from a thermodynamic point of view, the system is completely characterized by a much smaller set of numbers. In this particular example, I need volume, temperature, pressure, either the mass or the number of particles, and maybe its chemical potential. But I also need an equation of state that relates these, and I need a state function. Now, which state function I choose depends on the canonical variables that I'm interested in, and this has been the bane of students in thermodynamics over the years. There are many different state functions that are defined for different situations. Depending on whether you're a chemist, you might be interested in enthalpy. If you're interested in steam engines, you might be interested in the Gibbs. If you're interested in constant volume or specified volume, you may be interested in Helmholtz. Entropy is a state function, energy is a state function. There's lots of state functions. The key thing is that in thermodynamics, the state of the system itself, the entire ensemble, is independent of history. And the prototypical example is in the upper right corner, where if I were to place that 2 gram or 1 mole of molecular hydrogen as a gas in partition 1, open the valve to partition 2, allow it to equilibrate, and then open the valve to partition 3, the gas would eventually equally distribute itself into all 3 partitions. But if I originally placed the gas in 2, then opened the valve to 3, then opened the valve to 1, I'll still get the same final state. In fact, I won't know where the gas originally was, whether it started in 1, 2, or 3, and what order those valves were opened. And again, that's one of the things about thermodynamics, is that it does talk about reversibility versus irreversibility, but we do lose that sense of history when you're talking about equilibrium thermodynamics. And many people have related this to time's arrow. And again, equilibrium thermodynamics has a set of basic axioms that have worked incredibly well. But because of that independence of history, the thermodynamic state of a system is independent of its context, except for the specification of its volume. And again, we have well-defined probability distribution functions for them. We can have in the first law, an energy balance equation, which is basically the difference between the coherent and incoherent part of the work, the incoherent part being the heat and the coherent part being the actual work being done by the system. And that's what relates the change in the energy state function. Second law gives us a definition of entropy, and so on. So again, equilibrium thermodynamics is a special case of a time-independent system in which its bath or its container it has been allowed to equilibrate. And so you're not looking at transient features, you're looking at that final state. But there are other states that the system could have, and these are the states away from equilibrium. But I'd like to point out that there are 2 ways you can be out of equilibrium. Being non-equilibrium can, in one case, be done because you can't reach equilibrium. That is, although you do have uniform thermodynamic parameters, the temperature is constant across, the pressure is constant across, and so on, but it's not in its lowest energy state. That is, the relaxation times of the system diverges and the system arrests in some intermediate state or at some sort of frustrated state. Perfect examples of these are gases, amorphous or disordered solids, and so on. But far from equilibrium typically is meant by a driven system. That is, there's energy flow. And there are states that the collection and the ensemble can attain only because it's being driven. And this is what we now refer to as active matter. In order to do that, you need 2 distinct thermal baths. You need an energy source and an energy sink. You have to have it flow through the system of interest, and the system has to be a finite size enough that gradients emerge. And again, in the simple case of a solid, the gradient that emerges is related to the thermal conductivity of the material. But the key thing is that energy minimization is no longer the criteria for defining that state. Entropy is not well defined. And in general, if things are chaotic and turbulent because it's not steady state, you can get all kinds of very active states of the system. But order doesn't usually emerge in those areas. It usually emerges when the energy flow and those gradients are steady state. And especially when there are internal degrees of freedom, such as found in liquids and gels. And I show a few examples of the surface of a vibrating drop in the lower left, actin filaments that are being driven by kinesin motors in the upper right, examples of being stuck in a not the lowest energy state of various ice crystals, collections of bacteria in the lower middle, and of course a cross-section of a plant stem. All of these things could have been active and driven, and then falling into a non-equilibrium state and simply being arrested. But these are what we really find to be most interesting in these systems. And of course, these are the characteristics. We really don't have a good idea about how self-organization, and again, how does this coordination occur without some kind of control mechanism, and what are the variables that are involved. So again, the emergent order happens on over many, many length scales, and we've seen many, many talks through this wonderful conference about that. What I'd like to focus on is synthetic biology. This is an example of a soft matter system that is distinct from cell engineering. So think of it as looking at the basic unit of biology, the living cell. That's like an engine. And in cell engineering, you're taking an existing cell and performing some kind of genetic modification or some kind of other replacement of parts of the machinery of it. And this has been around actually for a very long time. The term synthetic biology goes all the way back to 1910 and 1912. But it really was only in the 1970s and 80s with the discovery of the DNA structure that genetic engineering could emerge. And it was in 1978 when the first synthetic human insulin, Humulin, was developed. But the real thing that broke the field open was the ability to have advanced microscopy, to not only see a cell, but see inside a cell, and the ability to control what's going on within individual cells, and even probe mechanical properties within a cell. So visualization and controls throughout the 1990s, along with a lot of really sophisticated synthesis techniques, allowed for a huge advancement in the other way of thinking about synthetic biology, of being able to take pieces that were never part of a cell, synthesize them yourself, and put them together. And we've again able to put synthetic biological circuits. These are the chemical sequences that form the signaling pathways within a cell, and make completely artificial ones and implant them into E. coli. And then, of course, just last year, the first artificial bacterial genome was developed. So there are two ends to this. As I mentioned, cell engineering is taking what's already there and changing some of the geometry. But on the other end, from physics and chemistry to build up the elements of a cell to the point where we could actually conceivably put a cell together from scratch. So there's a lot of, again, enabling technologies that have allowed that. At the bottom left, I show you the top 7 protein, which is the first protein designed for a particular fold that was not seen in nature. And because of these advances in microscopy and microfluidics, we have a really good idea now of how the ribosome actually synthesizes. So I show in the lower right the various processes that are involved in creating some of these synthesis machines, these little biomachines. We now know enough to be able to put together all of these pieces ourselves and even substitute different aspects of this machine. And again, this is quite a tour de force of a combination of physics, chemistry, and biology. There are various applications. People are thinking about doing not only changes within a biological system for medical applications like biosensors, but even designing things like synthetic life or biological computers. And again, there are a lot of open-ended questions. If you have this ability to now control not just the membrane, not just using colloidal physics to create a cell-like structure, a micelle, but you have compartments within it. And then interspersed within the compartments, the cytoskeleton. You can attack certain problems, and I'll just point out 2 things. One is the problem of chirality. Everything that we have in the cells in our bodies and on Earth are right-handed. So this is a big question because most of the molecules exist both right and left-handed. Why did nature pick one-handedness for this? So that's a a question. But there's also, begs the question, if there are left-handed versions of all the molecules that make up a cell, could we boot up a left-handed cell? Would it work? What would it do? We do know that there are major changes that can happen between different handiness. A typical analgesics you can buy at the grocery store is a left-handed version that works very well, clearing your nose. The right-handed version is an illicit drug, methamphetamine. There's other questions. There are over 35,000 variations of lipids. These are the fats that typically are interspersed and intercalating between the cell walls. And it's not clear why there's so many variations of these lipids. What role do they have at the interfaces? But we do know that these things can strengthen or weaken cell walls, perhaps for some various purposes. But what if we were to make artificial lipids? Lipids that allow us to connect directly across a cell wall and access that machinery directly. So again, synthetic biology allows us to think of some very important questions. But these are all collections of things that are out of equilibrium, and they have to be actively out of equilibrium. In other words, there has to be an energy flow into and out of the system. And of course, all of these things can have practical effects. We could have artificial systems that can mimic the creation of materials that make seashells to make bioconcrete. It's remarkable how incredibly carbon intensive concrete is, the way we're doing it now. But biology has been able to create calcium carbonate under very low energy conditions, under ambient conditions. We would have a tremendous impact on our climate and mitigate many of the changes if we could just simply create concrete the way biology does. And of course, there's implications in information complexity, and of course, biomachines that we never even thought of, creating things like biofuels, fabrics, and using materials that are found. So some of these things are found in locations that we haven't yet visited it. So some of these things are discussed in a recent special issue of the MRS Bulletin. And again, just to reiterate what's been said in many of the talks here, I thought I'd include the fact that this occurs across length scales. And of course, people have been more ambitious in their slides and have included galactic and cosmological length scales. But here I'm just looking at things that are essentially tabletop. How we can get simple things like hydrogen and carbon to create a molecule. That molecule, maybe by accident, could occur in nature. This particular one is my favorite. It happens to be caffeine. But nowhere in this molecule can you see where it has the caffeine effect. That's a biological context. But all of these molecules and all the various syntheses come together with the basic unit, the cell, cells forming multicellular structures and multicellular life forms, can form hierarchical structures like the swarming and flocking behavior. And of course, ourselves are the best examples for these kinds of things. So the key thing is that there's the whole arc of this research is quite broad and quite confusing. And so there's a need to use some of this new technology of being able to probe and get physical information on these kind of ordering systems that might shed light to some of the theories that are being developed. And so as an experimentalist, one of the simple examples that I've been fortunate enough with my student, Atanu Chatterjee, who just graduated, and very proud of him, who will be soon joining the Weizmann Institute in Israel as a postdoc, to take a very simple, well-known system, but with the ability to do some detailed microscopic, not quite nanoscopic, but quite new measurements of what's happening in such an ordering system that is far from equilibrium. So the old system that we hope to breathe new life in are simple Rayleigh-Bénard convection cells. This has been known since 1916. Its interest is mostly in fluid dynamics, and a lot of work has been done to explore the fluid mechanics of such systems. But if we take a step back and realize this is an example of an ordering system driven by an energy flow, we might be able to use some modern techniques to get some additional information from this in terms of ordering. So just to remind everybody what it is, you have a thin fluid layer. You have your two baths. The bottom bath is where the heat is applied. The top bath is where the heat escapes. The fluid is thin enough that a gradient will form. And because of the internal degrees of freedom, convection will occur. Because of the expansion as it heats, the fluid rises from the bottom to the top. Where it cools, it then becomes more dense and heavy and falls. And this pattern, this convection cells will then form patterns within the fluid film. But the cool thing that can be done now is that we can take thermal images of these systems and ask questions like, what are the statistics of the temperature variations within these convection cells? And then relate those to ideal cases as if there were no convection, pure conduction was occurring. And here are two examples of some images that we had taken on a thin fluid film. You can get hexagonal cells to start forming, you can get roll patterns to start forming. You can play around with the variables of thickness and viscosity and the amount of heat being applied to the system to achieve any of these types of ordered states that only occur because heat is flowing through the system. And so this is one example of an active matter. So what can be done with these images is you can again take them as a function of time once the heat is applied and watch the onset of the order. And you can take them at various thicknesses under various power loads. These images can then be analyzed. So we can do time and space analysis of the statistics of the thermal profiles. So again, we can look at one spot on the pattern as a function of time once the heat is applied. Or we can look at a given point in time, the distribution of hot and cold spots, as well as near the edge where it was uniform in temperature because of the sidewalls, the heating by the sidewalls. So we had a way of comparing directly the statistics of the temperature variations pixel by pixel among all of these different structures that form. And so some of the results are kind of interesting. If you were to do just the histogram of hot and cold pixels on the left, the x-axis is temperature. If you vary over the entire region, over the hot, you can see the hot and cold peaks. If you then focus in on the statistics of each hot and cold peak, you can see that they're not exactly Gaussian, but they can be described as such. But more importantly, we can look at the standard deviation of these patterns as they form. So on the right is the time axis. Once the heat is applied, you have at the top what happens when you don't have enough heat and no pattern forms. The system just simply increases in its temperature, the standard deviation rises and then plateaus. But when you have a pattern that forms, it initially rises and then falls. The standard deviation falls to some point in time once the pattern has stabilized, and then the standard deviation continues as a plateau. So it's a unique feature that the statistics of the thermal profiles give us a signature of when pattern formations occur. And again, we've used this to think about in terms of the cycles within the system, how pattern formations can lead to different distributions. But heat in, heat out is being partitioned within the fluid where coherent work is emerging, a pattern is forming, but at the cost of some incoherent work or heat being redistributed So again, nothing is violating the second law. The rest of the universe will continue to increase its entropy, but internally, it appears that the entropy that is decreased by the pattern is balanced by the work it took to create those convection cells. So what I hope to show in this simple example is that there's a lot of effort that can be done on tabletop experiments to probe and even test some of the theoretical ideas that were presented in this wonderful conference. What I'd like to do in this last third, and I think I have about 6 minutes left, is talk about how we navigate this research. So this is where I'm changing my hat to being a program director at NSF, and how we're thinking about, and how a lot of funding agencies are thinking about this kind of work, where you're looking at disciplines that are cutting across, or you're looking at phenomena that are cutting across many disciplines. The first thing to realize is that we are thinking about research paradigms in the funding agencies as a way of differentiating the sort of the role each agency has. So the NSF's primary mission is for discovery, just for discovery's sake, that will eventually have an impact on the economic and social health of the country. But there are specific agencies such as NIH or the Department of Energy that are looking at mission-specific needs of the country. And so while we are on the discovery end, the other agencies are more of the development end and perhaps the deployment of some of this research. What we'd like to do in all of the agencies is ask, as we're probing, especially in the synthetic biology realm, that whatever we do, whatever we put out in terms of new materials, new devices, new chemicals, that we think about the degrading and decommissioning of those materials, how to recycle them, how to close the loop of those materials. And this is, again, a brand new effort within NSF. There's a meta program called Sustainability that looks at how we decommission or degrade materials either for recycling or repurposing. Now again, this is a very complicated space. It doesn't go just around the corners. Sometimes discovery can be done specifically for deployment, and sometimes you develop things specifically for decommissioning. But these are the things we like researchers to think about in terms of where their research lies. Are the questions that they're asking of a fundamental nature, or are there questions asking of a more of an applied or engineering nature? But in terms of this research in complex systems, in emergent order, it's very complicated. And so we've taken the approach of what's called a square table. So most people are familiar with the phrase a roundtable meeting. Those are where you want many, many different viewpoints of a particular topic. But there are many aspects of this research in which there are not particular topics involved. There are many ideas that are coming together, and so some structure needs to be implemented in order for that conversation to be effective. So again, our idea, we've done several of these types of meetings, is to bring together two communities that don't normally talk to each other, along with the funding agencies and the regulatory agencies, to have meetings to talk about very specific things that they can do together. And this builds capacity of interaction among the groups. And finally, I'd like to talk a little bit about convergent research and my particular view of it. So we've mentioned many times, both in a social context and in the research context, that we need to bring together physics, chemistry, engineering, material science, mathematics, computer science. And each one of these disciplines are like fruits. And what we mean by multidisciplinary is a fruit bowl. The disciplines are well-defined. They have clear edges and connections between them, but they're not a lot. Interdisciplinary is a fruit salad. This is where things are more finely chopped, but you could still pick out where's the physics, where's the chemistry, where's the math, and so on. What we really want is a smoothie. This is convergent research. This is where the different aspects of physics, chemistry, math come together to something new that you can't pick apart and have those clear boundaries here. So whenever I ask people about their interdisciplinary nature, sometimes I will ask them, are you a fruit bowl or are you a smoothie? And finally, I'd just like to give you a sense, since I was working in the condensed matter physics program of the Division of Materials Research, you can find this information online at the nsf.gov website to give you a sense as to not only the soft side and what can be done, but also on what's traditionally called the hard condensed matter side. And here are just some examples of solid state spin chains that are out of equilibrium, phonon control and iron-based superconductors that are out of equilibrium. Every paradigm for quantum computers requires that it be driven out of equilibrium, and then its computation, the mixing of those quantum states, occurs as that system relaxes back to equilibrium. In soft condensed matter, we have things like topological solitons that can show up in liquid crystals and colloids, dissipation mechanics in soft molecules, mobility of and interactions and order in active granular systems and chiral fluids and like. And I'd like to end by just giving you one little treat from one of the PIs that I was lucky enough to work with as an example of two kinds of active matter. The first is on the left, these actin filaments driven by kinesin motors, and the energy source is ATP. And as long as there's ATP in the system, you can get this kind of pattern flow. And that has been referred to as active mixing or active pneumatics, because you can now keep track of all of the vortice points and bend points and really probe the statistics of those systems. You can also look at a colloidal system that under temperature quenches, you can get reversible and dramatic changes. So on the right, there is a droplet of an elastomer with a liquid crystal interior, and you're not looking at it under cross polarizers. What you're looking at is a structural change that will occur under a temperature quench, where this droplet will start forming fingers that will twist. And as you reverse the temperature quench, this thing will go back. And so now we're looking at, as I said, new states of matter that are driven out of equilibrium. And these are things that I hope you found interesting. And I'd like to, again, thank the organizers for the opportunity to talk to you. So I think I'll end there. And I guess I'll stop sharing my screen. And I don't know if I left enough time for questions, so
You did. We have, the session goes, if I'm not mistaken, around until 10:30, and so we have about 13 or 14 minutes for questions on all of the talks.
That's true, Bruce.
So I'll open up the floor now.
This is Carrie. I have a question for Germano. Thanks, that's very interesting. You mentioned, in your mention of looking to biology or biomiminic ideas for making materials, it seems to me, so you can correct me or clarify, that, a lot of, biology is kind of making, building up shells, for example, atom by atom to some degree, and our industrial processes have somewhat subverted that. We just throw a bunch of stuff together and make it real fast by putting in a lot more power. So do you really see, or how do people see that we could, say, mitigate climate change by finding low carbon processes that mimic biology, but that are much slower, and that our economy isn't really structured on doing things slower? We're trying to do things more quickly.
Of course. And so that's one of the areas of research is not that we don't know what those chemical processes are. The hard part about using a fermentation process is that you have to get the products out of the cells, and you'd like the cells to continue to produce these things. If you can get away from cell engineering and you can do synthetic biology, one thing that's being worked on is creating cell-free chemical processes. Basically, the signal pathways that created that particular chemical process scaled up to the size of thousands of kilograms without the cell wall itself. So there's a lot of machinery that the cell has, for example, for reproduction, that if we're interested in creating a biofuel, we don't need that part of that machine. So if we can solve that problem, then the scale-up problem that you're referring to can be solved. As you rightly said, we have these very energy-intensive industrial processes because we need massive amounts of these things. So for example, spraying an engineered bacteria in order to create a concrete reef near the shoreline, that'll take time to do. But in a week or so, I could pour the concrete and have a dock made. So there are going to be these limitations that occur, and they're also one of the reasons why we have this interest in decommissioning is that if we're going to release organisms that have been engineered for these particular functions, they don't respect international boundary lines in the ocean. They're going to go everywhere. So they have to have an off switch of some kind.
Yeah, can we make the off switch?
So clearly there are some applications that this will work well and can help mitigate things. There are other applications that, the more traditional industrial processes will be more appropriate.
May I ask from Professor Galavotti?
Yes.
So my question is that statistical physics analyzing far from equilibrium systems instead using close to idle systems. So why can one expect that with these specific arrangement or microstructure like oscillators, Anosov systems would lead to some universal consequence?
Yes, what I would say is that somehow the opposite is true. What happened in the last 20 or 30 years is evidence about motions which are done by chaotic systems and so on. And in the end, most of the phenomena that were observed have been reduced to properties that appear already in the systems that you are quoting. It's like you look at the planets, and then you discover the Kepler's laws. Then you look at the motion of a top, and you discover the Euler equations for the top. Then you go to the Euler equations for a fluid, and so on. And in the end, you discover that all the ordered cases that I have mentioned can be reduced to a paradigm which is essentially the harmonic oscillator. And on the contrary, when you study, when complex systems have been studied, first it has been, well, I still remember when I was a student that even if, well, actually, already the book by Landau-Lifshitz tries to reduce to harmonic motions the turbulence, the beginning of turbulence. And in the end, you end up by realizing that the paradigm for all these chaotic motions, complex systems, are the hyperbolic motions or Anosov systems. So it's rather the contrary. We have learned from the fact that there are some general properties common to many systems which apparently have nothing to do with each other that can be put in the frame of these very simple systems. So it's just physics always tries to find a simple paradigm to represent what is of interest. So I would say it's the contrary. It has become clear that the hyperbolic systems or the harmonic motions are paradigms for a large class of systems, of phenomena.
Do you have some general, let's say, maybe not strict, but some proof of that? like the proof of the equilibrium systems thermodynamic, the thermodynamics temperature of Kelvin?
No, in my talk, I've spoken only about stationary systems and in particular equilibrium systems. For them, so for equilibrium systems, there is no doubt about what temperature is. For non-equilibrium, I don't think you can define temperature of a non-equilibrium system. You can define the temperature of the thermostats with which it exchanges heat. But what is the temperature of a system which is heated by on one side and made cold on the other side is not defined. Perhaps you can define a local temperature, but if the system behaves as in equilibrium locally. But so that's it. That's it. What can I don't know what you're really asking more. Yes.
Maybe, may I ask a follow-up question to that? I'm familiar with the Sinai-Ruel-Bowen measure that's sometimes used for driven dissipative systems.
Yes.
And along with that comes a formalism that looks very much like thermodynamics, and it's called the thermodynamic formalism. To what extent is that helpful in accomplishing the program that you're setting out to accomplish? And where is it missing things?
Yeah, I'm not trying to convince about the worth value of a program. It's just that if you have had ever doubts, questions about why a system goes to equilibrium and what is equilibrium and so on, these things have been answered. I think the first remarkable thing is that there is no difference between equilibrium and non-equilibrium from the point of view of theories, because the unifying feature is the chaoticity or hyperbolicity of the motion. And then this implies, and that's really remarkable, it implies the uniqueness of the probability distribution that describes the statistics of the object. Maybe I can, well, to say about a universal consequence of these chaoticity properties of stationary systems which are reversible, and as I said, reversibility has nothing to do with friction. You can have a reversible system which behaves as under friction, and it is perfectly fine. Let me give an example of one of the consequences which I found particularly shocking. So it's about the fluctuation relation, the fluctuation theorem. If you have a chaotic reversible system, and there are many examples of that, then you can ask what is the probability that the entropy production per unit time has a certain value, say S, compared to the probability that it has the value minus S. So let's go to dimensionless units. So let's say the average value of the production of entropy is 1. And then you can ask what is the probability that it is minus 1 in the system. Well, this is as a universal formula which tells you that the logarithm of the difference of the probabilities of the two events is just a linear function of the entropy production and of the time of observation. Now, okay, that's fine. Maybe you are interested in that or not. But then suppose you ask also, what is the probability that at the same time that there is an entropy change from rate from 1 to -1, you observe another observable, right? And which follows a certain pattern of evolution in time. Then you can ask, what is the probability that I see the opposite pattern followed by the given observable? Well, I find it is extremely remarkable that the ratio of the probabilities of the forward and backward evolutions of this observable and of the change of creation rate of entropy, this ratio is independent of the observable that you observe. So let me say in a pictorial way, if you want to see the Niagara Falls go up instead of down, the only thing you have to do is to change the sign of the entropy that is produced in the process, which is something I don't recommend that you even try to do. But it is, I mean, when you study small systems, microscopic systems, this kind of information may be of interest, practical interest. So that's an example of a universal relation, you see. The probability that a certain observable goes in one way and goes the other way is independent of the observable, provided at the same time the entropy does the same. Entropy creation does the same. And that is, well, it's universal because it does not depend on the observable.
Okay, can I ask a question from Dr. Iannacchione? It relates to the Rayleigh-Bernard system, and I was fascinated by the variance, the standard deviation going up and then basically making these non-monotone trends. And I was wondering what controls that. So are there any factors that you can use to increase or lower that variability in the system? And I'm interested in this because in my lab, we built synthetic gene circuits that control cell-to-cell variability, living cells. So I was wondering how that works in this Rayleigh-Bernard system.
Well, that's a very good point, and this relates back to the discussion that was just had about one part of it, about temperature. The thermal cameras allow us to probe local temperature very fast. The camera that I was using had 100 frames a second, and you're not waiting for equilibrium between that and a thermocouple or a thermometer of some kind. So these local probes of temperature allow you to map the temperature profile. So it's a manifold. In equilibrium, that manifold is flat. And out of equilibrium, it takes work to create these gradients, thermal gradients within the system. And our analysis was that there are additional information when you look at the statistics of the pixel to pixel variations of these things. So for the standard deviation to drop when a pattern forms, it means that each one of those pixels are fluctuating less than they were before. So as the heat was applied, the temperature was rising, no pattern was formed, you saw lots of these little variations. Now keep in mind, this is limited by the frame rate of the camera. And as technology progresses, we'll have better and better statistics, both in terms of how small a region we can probe and how fast we can probe. So this is, this work is just really a slice of the time domain. And of course, the pixel size gives us only a small picture of it. So in your particular case, if you had a thermal camera that was accurate enough in terms of pixel dimension, even at 100 frames a second, most of the processes are slow enough that you would be able to follow those processes. And what that drop in standard deviation may mean is that there's a cost once the pattern forms. And again, it's tempting to take that cost as a drop in the local heat variations, local heat gradients in the system. And that's why at the last slide of that middle third, I suggested that as heat flows or any energy flows through a system in gradients form, if the system can respond internally, it will partition that into an increase in the amount of work being done, but that has to come at a cost. there has to come from somewhere, and it may come from the thermal fluctuations in the system. So even though averaged over the entire system, and this is something I didn't mention, but if you were to average the temperature over that entire surface, it's exactly what you'd expect if it was just heat conduction. The convection does not create more efficient transfer of energy from one side to another. All it does is it takes this manifold, this flat temperature profile, and creates a dimpled surface.
Excellent. Yeah, thank you very much. And it's interesting that even in the stationary state, you have a variance persisting, apparently.
Oh, sure. The plateauing of the increase in plateauing of the standard deviation is simply because you're going from room temperature at 20 degrees C to something like 70 or 80 degrees C. So that's just simply scaling the root mean squared fluctuations in the system. It's the drop that's the most dramatic part. So again, it is limited by the bandwidth, essentially, both temporal and spatial, of the camera, but it allows us to now interrogate these thermal variations within the system.
Thank you very much.
Thank you very much, everybody. I'd like to thank our three speakers. Fascinating session. We're a few minutes over now, so we should probably end it. Thank you very much. It was riveting, and thanks to the people who asked questions as well, the thoughtful questions.